Morning, everyone. Thank you for joining the webinar. I am the moderator and co-speaker of this webinar. With me is our guest speaker, Shidroy Tripathi. And before we get started, a few housekeeping items. The two talks today are pre-recorded. So after this, we will play the two recordings, and then we will have a live Q&A at the very end. This webinar is being recorded. After this webinar, in a few weeks, you should receive the recording in your email. You are all muted as attendees. At any point in time during this webinar, if you have questions, please use the questions box that's in your GoToWebinar on the upper right corner to type in questions at any time. We will answer them at the very end, unless it's a question related to logistics or being able to hear the audio in the webinar. You'll also see a tab with the materials. These are some materials that we think would be relevant for you if you're interested in this webinar. And finally, when the webinar is over, you will see a pop-up for a survey. We really appreciate if you can give us feedback through the survey. That's all. And with that, please enjoy the recorded talks first, and I'll see you at the end for the live Q&A. Good morning, everyone. Thank you for joining this webinar. My name is Liz, and I'm the Associate Director of Product Marketing at PacBio. I'm going to give you an intro talk first about full-length bulk and single-cell RNA sequencing with Kinnex kits. RNA is a critical driver of proteoform diversity because alternative splicing, along with genetic variants and post-translational modification, generates a vast spectrum of unique protein forms. Understanding RNA isoform differences is essential for defining the full functional complexity of the human proteome and unlocking insights into biological mechanisms and disease processes. Traditional short reads have trouble assembling a full transcript, and this is exactly because alternative splicing produces many RNA isoforms that uses the same exonic structures. In contrast, with long-read RNA sequencing with PacBio, the Iso-Seq method, we sequence the entire full-length cDNA. No assembly is required. That is the case for bulk RNA sequencing. In single-cell RNA sequencing, short reads can only sequence either the 3' or 5' end of a transcript. This is because of the limitation of needing to record the single-cell barcode and UMI information. Regardless of how much it's sequenced with short reads, you can only get gene information for single cell. In contrast, with long-read RNA sequencing, you can get the entire full-length isoform information plus the single-cell barcodes and UMIs. The more you sequence, the richer information you get. An example of using long-read RNA sequencing to improve mass spectrometry is in this recent publication. Researchers integrated Iso-Seq data with targeted mass spec using a method called Tomato. They achieved a 3.6-fold increase in isoform detection and identified previously unannotated proteins. This approach strengthens confidence in proteogenomic discovery, enabling more accurate validation of alternative proteoforms and laying the groundwork for new biomarker discovery and functional studies. A lot of this RNA work is enabled by the recent Kinnex kits. Kinnex is a general amplicon concatenation method for throughput increase on HiFi systems. This is because HiFi reads are best for DNA fragments that are 15 to 20 kilobases long. Most cDNA or GDN amplicons may only be 1 to 2 kb long. By concatenating them in an ordered array and sequencing with HiFi, throughput for each cDNA can be increased. There are 3 Kinnex kits: the Kinnex single-cell RNA kit, 16S RNA kit, and the full-length RNA kit. They offer different target throughput based on different concatenation factors: 8-fold, 12-fold, and 16-fold. Choose which kit is appropriate based on the average amplicon size of your application. We've tested the sequencing depth for bulk RNA sequencing using Kinnex kits. Looking at across different samples and tissues, we find that generally for most samples, somewhere between 10 to 20 million reads are when 80% or more of the known genes and isoforms are identified. Another way of looking at this is looking at the isoform discovery curve on the right-hand side. At 10 million reads per sample, one new isoform can be discovered for every thousand new reads. Based on this, this is what we recommend. For isoform discovery and comparison of moderate to rare transcripts, such as most human genetics disease studies, where there are disease versus normal samples, 2 or 3 replicates each, we recommend a 10 million read per sample multiplexing strategy. For isoform discovery and comparison of highly expressed transcripts, such as looking for highly expressed targets or large cohorts, we recommend 5 million reads per sample. For plant and animal genome annotation, it depends on how many tissue types are available. We recommend either 5 million reads or 10 million reads per tissue. To summarize, Kinnex concatenation enables throughput increase by 8, 12, or 16-fold for amplicons, including bulk cDNA, single-cell cDNA, 16-SGDN amplicons, and more. By enabling sequencing full-length isoforms, one can discover more novel peptides or discover novel isoforms for biological and disease studies. To learn more about Kinnex, visit pacb.com/Kinnex. We also have bioinformatics tutorials and datasets at Iso-Seq.how and pacb.com/datasets. Thank you for listening to my talk.
All right. Really excited to give this seminar for PacBio on our work using PacBio's latest Kinnex system to study alternative splicing and neurons. So I'm Srijoy Tripathi. I'm a scientist at the Krembil Center for Neuroinformatics at CAMH in Toronto. I'm also an associate professor at the University of Toronto. I'm about to present the work of a lot of people. So this project was led by Nuo, a PhD student in my lab, and Catherine Reinhard, a PhD student in Craig Smybert and Howard Lipschitz's labs. This is the work of a lot of people. Big shout out first to our funders, Safari, particularly Alan Packer, who gave us the opportunity to basically do a lot of long-read RNA sequencing on these samples, and then our PacBio accounts manager, Grace, who's been amazing. So as we all know, most genes have many mRNA splice isoforms. So for example, a precursor mRNA is spliced into multiple mature mRNA isoforms. So here you can see that this blue exon here is spliced into the isoform on the right, and it's not included in the isoform on the left. And then when that's translated, that can result in different proteins that then can give, and these different proteins can have different functions. And so this is what we're trying to study when we're trying to assess splice isoform diversity using RNA sequencing data. So here we conducted a very deep functional genomics set of experiments to understand neuron development in great detail. So we're using a classical system called the NGN2 system for basically growing up human neurons in a dish from iPSC cells. And then from this system, we have basically three types of data we're collecting. We're collecting super deep long-read RNA sequencing data. I'll tell you more about that in a sec. And then we also have paired short-read RNA sequencing data and shotgun proteomics data. But really, like the linchpin data set of this talk is our long-read RNA-seq data. And so, why did we do the experiments? What are we hoping to learn? So first, we wanted to build a comprehensive atlas of mRNA isoform diversity along this process of neuron development. Second, we then wanted to validate our splice isoforms at the protein level using our proteomics data. Third, we wanted to quantify the landscape of alternative splicing, and by asking, which isoforms are used at these different time points. And lastly, we want to ask, does all this matter? Does this matter for, human disease risk? Okay, so as I mentioned earlier, we're using, here in this project, we're using Kinnex. That's the latest PacBio chemistry kit for long-read RNA-seq data. So here on the left, I have a screenshot of this paper from Patawari et al. They have a very similar experimental design to ours, but they're using tissue from from fetal developing brains. We love this paper. Like, it's basically, our study is very much inspired by this paper. But just to motivate kind of what you can get out of Kinnex. So on the left, we're just plotting, total read count per sample from this prior paper from Patawari et al. And that was collected using PacBio long read RNA-seq, but it was collected, sort of in the era pre-Kinnex. And with our data set, we're able to get so many more reads per sample. So, it's about like a 40-fold increase in read depth. And so in total, we have 465 million QC passing reads. So between 25 to 40 million QC passing reads per sample. So we're really, really excited about this data. We think that we might have collected among the deepest long read RNA-seq data sets to date. Liz can confirm that maybe at the end of this talk. Okay, so so we did a bunch of this deep long read RNA sequencing. Like everyone before, we basically we look at our long read transcripts and we compare them to GenCode, and then we're basically classifying our transcripts into a bunch of categories. But basically, we have a category for known transcripts. So these are transcripts whose junctions match exactly the same as GenCode transcripts. We call that full splice match. And then we have three categories of novel transcripts: incomplete splice match, and then novel in catalog and novel not in catalog. I didn't come up with the names, but basically these are based off of comparing the junctions observed in these isoforms to those in GenCode. So then, by looking at the isoforms that we got back from this data, we see that the majority of our splice isoforms are actually novel. So about 55 to 60% of our splice isoforms are novel, meaning that they're not present in GenCode. Because we have such deep data, we can actually be somewhat stringent in what we call a isoform what's not a splice isoform. So here we're defining novel splice isoforms as those that are present in at least 5 reads in 3 or more samples. So again, like, we're only using transcripts that we're observing multiple times across multiple samples. So maybe as you'd expect, a lot of the novel isoforms that we're discovering tend to be those that are expressed more lowly. But many of these novel isoforms, so here in orange, in these 2 different colors of orange, they're present at least 100 to 10,000. Yeah, 100 to 10,000 reads in our data set. So again, they're lowly expressed, but they're still quite abundant in our data. Okay, so we now collected this. So to validate our splice isoforms, we turn to our short read RNA-seq data. And so specifically, we're looking at splice junctions that we can detect in our short read RNA-seq data. So a splice junction is defined as a read that spans the boundary between 2 exons, and then they can be assessed without any controversy in our short read data. And so we can basically look at our short read data and ask, well, are the splice junctions we observed in our long read data also present in our short read data? And so, then looking first at the known splice junctions, so these are splice junctions that are present in GenCode 47. Virtually 100% of the known splice junctions that we observed in our long read data are also present in our short read data. And then turning to our novel splice junctions, so these are novel splice junctions that are not present in GenCode. Around 80% of those are also validated in our short read data. So we're really happy with this. We're really happy with this, and there's just the high degree of concordance between our long read splice junction and our short read splice junctions gives us a lot of confidence in our novel long read RNA-seq data and this novel isoform atlas that we've built. So then moving to our proteomic validation. So now we want to validate, we want to use our proteomics data to validate the translation of these novel splice isoforms. So, probably doesn't need to be said, but I'll just say it. So, RNA is translated into proteins. And so just because we can observe all of these novel isoforms in our long read data, that doesn't necessarily mean that they would be translated into proteins. And so that would be, we want to see if some of these are actually validated at the proteomic level. Okay, so the first question that we need to ask is, well, which isoforms actually have the potential to be translated into new proteins? And so, with our long read data, we just get mRNA isoforms. We don't actually know, we don't know beforehand how are they translated into proteins. So what we did is we applied a tool called ORF prediction. So ORF prediction, ORF stands for open reading frames. And so that basically identifies the part of the mRNA isoforms that have the potential to be translated into a protein. So we're using a tool for open reading frame prediction called Orphanage. Again, I didn't come up with a name, but it's a recent tool for open reading frame prediction. We think it's sort of best in class today for this task. And so from our set of almost 200,000 splice isoforms, 162,000 of them have the potential to be translated and have open reading frames. And then because different open reading frames can actually code for the same proteins because they can have different UTRs, we have a set of 81,000 unique ORFs that have the potential to be translated into proteins. And so this set of open reading frames then are the input to this next part of our project. So as I mentioned, we have mass spec data, and then we can use this mass spec data to validate our splice isoforms at the protein level. And so if you're not familiar, the way that this mass spec and the way that proteomics works, or mass spec based proteomics works, is that you take your long proteins, you cleave them using trypsin and a protease, and then you get basically these short peptides. And then you can then look at the amino acid sequences in these peptides via your mass spec. And so we're using an approach for this type called long-read proteogenomics. I have a citation here. This is a paper from Gloria Schenkman's group. And so basically the way this works is you can then go, so here we have, basically two gene tracks. On the top, we just have a collapsed version of all transcripts for this gene ZMYND8. And then on bottom, we have a novel transcript in this gene with a novel starting exon, and that actually codes for a new protein. So we have this novel starting exon. And so that we can go looking at our mass spec data. So this is the spectrum that comes out of a mass spec. And then this sequence of letters actually corresponds to an amino acid sequence. And so this amino acid sequence actually corresponds directly to this, kind of corresponds one-to-one to this novel transcript here and this novel part of this transcript. Therefore, this is confirming the translation of this novel transcript. And so we can do this process for all of the proteins in our data set. And so we can then ask, just like we did for our short read data, what percentage of our known and novel proteins are validated by our mass spec. And so here we see that 15% of all possible peptides, all possible known peptides, are validated by our mass spec data. And correspondingly, 1.1% of our novel peptides or 1.1% of all possible novel peptides are validated in our mass spec data. So that 1.1% corresponds to 160 peptides, corresponding to 440 proteins and 151 genes. This may or may not sound like a large number to you, but this number of 160 peptides is comparable to recent papers that have tried to do a similar strategy of using mass spec to validate the translation of novel isoforms discovered in long-read RNA-seq data. Okay, so moving on to the next task, so alternative splicing. So the first thing we did is we looked at differential transcript expression. So basically, our between pairs of time points here. So I'll say a little bit about the kind of data we have. So we have basically 3 time points of data. We have time point 0, time point 4, and time point 30. The numbers correspond to the numbers of days in vitro that these neurons or that these cells are allowed to grow up. And at time point 0, that's iPSC cells, and time point 30, that's mature neurons. And time point 4 days in vitro is something in the middle. It's like intermediate cells. So between And so we see the most amount of differential expression between time point 0 and time point 30, corresponding to about 35,000 35,000 differential transcript expression events. I should say here, we're just using our long-read RNA-seq data here. We're not like using our long-read. We're not using our we're not even looking at our short-read RNA-seq data at all for this. So we're just looking at differential expression with our long-read data. And so most of or about half of these differential transcript expression events are in our novel isoform categories, ISM, NIC, and NNC. And then, beyond differential transcript expression, which is basically like, just absolute or total expression levels between pairs of time points, we can also assess isoform switching. So isoform switching is like when you have, increased usage of one isoform relative to another. So for example, at our iPSC time point, you could imagine that isoform 1 is the dominant isoform for a gene. But at the neuron time point, at the mature neuron time point, the isoform usage could switch. So now that isoform 2 becomes the dominant isoform compared to isoform 1. And so this type of isoform switching analysis is called differential transcript usage. We see over 15,000 total differential transcript usage events by looking at our long-read data. And so between time point 0 and time point 30, about 40% of our isoform switching events are corresponding to novel genes. Just to provide an example of what this looks like. So here I'm just focusing on one gene, AGO1. This is an autism risk gene, or this is a bona fide autism risk gene. This gene has 3 primary isoforms, which I just labeled conveniently A, B, and C. A and B have the same, protein structure. They have the same CDS, but they differ in their 5' and 3' UTRs. And isoform C is similar to A and B, but it's missing this 3rd exon, and then it also has different UTRs. And so because it's missing this exon, our open reading frame calling tool then thinks that it predicts that the open reading frame actually starts here in the 4th exon. Okay, and then I'll say a little bit more about this in a sec. So we have these 3 isoforms. The first 2 isoforms, A and B, are known, meaning that they're in GenCode, and this 3rd isoform is NIC, meaning that it's novel. And so we can look at total gene expression of this gene. In this gene is not differentially expressed between time point 0 and time point 30. There's no change in the overall gene expression. But then when we look at the isoform usage of these 3 isoforms, we see that at time point 0, the isoform, at time point 0, basically like these 3 isoforms are expressed more or less equally. But at the neuron time point, T30, most of the expression of this gene is the 3rd isoform C. So again, like these, between gray and black, this black bar up here, becomes greater. And so this is an example of an isoform switching event where in the neuron time point, most of the isoform usage is isoform C. So then looking in detail about the changes here, and then thinking about the structure, what these different transcripts do. So, well, the difference, so this is now all, my summary is all with respect to the neuron time point. So for the neuron time point, then, the isoform that's more expressed in neurons tends to have a longer 5' UTR. So that's shown here in pink. They also have a longer 3' UTR that's shown here in green. And then also in this neuron time point, then isoform C, it's missing this, because it's missing this 3rd exon, then it's basically completely missing this critical blue domain. And this is an ARGO-N domain, and this is this is like part of the essential function of this gene. So so we call this domain, or this type of isoform switching event would be called a domain loss. This is the loss of a critical protein domain. So in general, across all of the isoform switching events that we've detected, we see lots of examples of changes, significant changes both to the non-coding structure of the isoforms, like their UTRs, as well as the, like changes that actually change the proteins like pretty drastically. So I've already highlighted on my prior example, I've highlighted 5' and 3' UTR changes, as well as domain gains. Some examples which I'll briefly highlight here are, in neurons, we tend to see a greater intron retention. So that's basically, where you have, an intron is retained between two exons. So intron retention is often associated with nonsense-mediated decay. So nonsense-mediated decay means that you have a premature stop codon. Um, and so this is really, you could imagine, or like we don't quite know why, but like neurons tend to include more introns, and then they tend to be those transcripts then would be subject to nonsense mediated decay. And then I'll just highlight again this example of like drastic changes in protein structure. So in neurons they tend to be like have they tend to basically be skipping exons that then lead to losses of protein domains, and then they also lead to losses of intrinsically disordered regions. And then this has drastic consequences for the total protein structure. Yeah, so again, this is pretty, to our understanding, like a lot of this is novel, and we don't quite understand why why is it that neurons are why are they tending to retain more introns, and why is that leading to nonsense mediated decay. um, One thing that we realized as we were doing this alternative splicing analysis is that we could actually use our long read data to assess the evidence of coordination between different types of alternative splicing. So here in this example of these two isoforms, isoform 1 and isoform 2, the first isoform has a short polyA, like basically a short 3' UTR here, shown at this this first polyA site 1, and then isoform 2 has this longer polyA site here. And in my example from isoform 1, this one contains this red exon here, which in my example is an alternatively spliced cassette exon, whereas isoform 2 does not have this alternatively spliced cassette exon. And so we wanted to use our data to see if we could find examples where we have like potential long range coordination between a poly-A site and an alternatively spliced event anywhere else in the isoform transcript. And so we're using an approach, or we're using a statistical approach developed in this prior paper published in NatComs in 2023, but used targeted long read RNA sequencing. And so we wanted to kind of take the approach of this paper that used this targeted method, and then kind of apply it to our untargeted, very deep long read RNA-seq data. So just to illustrate just what this analysis looks like, I'll turn back to this example of AGO1. So that's the same gene that I showed you a couple of slides ago. And so first, so here, this is this is a read coverage plot for AGO1 at time point 0. So in these first 3 rows, this is the read coverage plot. And you might be asking, well, this doesn't look like a read coverage plot, but this is what a read coverage plot looks like for long read RNA-seq data. It's basically all of the transcript is present in every read, like you'd expect. So the way we've kind of summarized this is we've summarized it to 3 poly-A sites. So this is poly-A site 1, poly-A site 2, and poly-A site 3, corresponding to my ABC isoforms before. And so and down below we have the the gene tracks, and we've highlighted in red this this alternatively spliced exon. And so we see for the first 2 poly-A sites, like the short and medium length poly-A sites, that this isoform in red, or this exon in red, is always present, whereas for this this longer poly-A site 3, this isoform in red is never present. Oh, and I should mention up here, these numbers here indicate their read coverage. So, for example, for poly-A site 1, we have, in this example, we have 293 reads for this first poly-A site. And so we can then quantify the inclusion of this red exon just using a standard percent spliced in analysis. And so these numbers here indicates that the exon 3 is always included for isoforms that include polyA site 1, and then 0 or close to 0 indicates that the red exon is never included when the reads or when the isoform is using polyA site 3. And so in these different colors indicates our 3 time points from iPSC all the way to neurons. And so what you can tell from this plot is that across the 3 time points, whenever polyA site 3 is used, exon 3 is never included, and vice versa for polyA sites 1 and 2. And so this is, this kind of it's kind of wild that you have coordination between the splicing of this third exon here and this poly-A site that's really, really far away. So just in general, we found like 3,000 total. So we were able to assess this in genes where we had both an alternative spliced exon and an alternative poly-A usage event, and so we found 3,000 such genes with both of these, and of these 3,000, 1,000 showed evidence of such coordination. And so here we just have some summaries of that. So first, over here, we have basically the distance between the 3' splice site and the proximal poly-A site. And so, this coordination, to me, it seems like it goes very far away. So this is in distance of unspliced or, pre-spliced mRNA. And so the median is between 10 to 100,000 bases away. And if you were to sort of just count this coordination up in terms of numbers of exons, it can be as far as, say 40 exons away. This begs an explanation for, what exactly is doing this regulation. We have some ideas, but this is something that, the field needs to explore, needs to look into further, beyond this. Okay, so for the last part of the talk, we're now going to switch to human genetics, and we're going to ask whether mutations in any of these novel isoforms might be deleterious. So, I've been talking to you so much about these novel isoforms and alternative splicing, but I haven't really said yet, do they even matter, right? So you might ask yourself, or a splicing skeptic might say, well, Srijoy, does it matter? Like, if you were to just completely, let's say CRISPR out the whole isoform completely, would the cell even care? We thought really hard about answering that question, and then we kind of landed on tools from population genetics. So what we thought to do was to use genetics or genomics data from large populations of control individuals to try to find evidence for negative selection in these novel isoforms. So the idea is that if they're important, they'll have evidence for negative selection in these large populations, and they'll tend not to be mutated in those populations. And then if they had lots of mutations in these control populations, that would indicate that, well, they're likely just not that important. Human individuals can tolerate having mutations in these regions of the genome. So what we did is using this large population database of Nomad, which has genetic variants available from almost 1 million sequences from control individuals. We basically use this data to get to find evidence of negative selection, and precisely we calculated mutational constraint. So what mutational constraint is doing is it's looking for basically is everyone in this population, do they all have the same base, or do they have different bases, and do they tend to have mutations? So first we looked at mutations in known isoforms, because that kind of provides a benchmark and baseline for what to look at for what we expect to see in novel isoforms. So first, we see that for mutations that code for nonsense, genetic variants that code for nonsense mutations in known isoforms, they tend to be strongly mutationally constrained, just as we expect. So control individuals tend not to have nonsense mutations in known isoforms, because if they did, that would basically completely disrupt that isoform, that would basically disrupt that gene, and so those tend to be under strong negative selection. And so the next strong, mutations, or the next strong genomic elements that are under mutational constraint, are elements that are in the essential splice sites. So essential splice sites are the two base pairs that are immediately flanking exons. So shown here in our schematic in gray. And so when you have mutations in the essential splice sites, then the splice site machinery can't identify where the splice sites are, and then it can't splice out the introns. And again, that leads to basically a completely non-functional isoform. And so these essential splice sites are also under strong mutational constraint. Yeah, and so and the way you get statistics out of this analysis is you basically compare the degree of mutational constraint of each element to the amount of mutational constraint of synonymous mutations. So a synonymous mutation is a mutation that actually codes for the same amino acid, and so synonymous mutations tend not to be mutationally constrained. And so that provides like a useful baseline for all of this analysis. And so again, this is for known isoforms. And then so to interpret the mutational constraint that we see in novel isoforms, we can compare that to what's seen in known isoforms. So the mutational constraint in novel isoforms is shown down here. We've replotted the mutational constraint in known isoforms as well. So first, I'll mention that the y-axis between these two plots is different. So in blue, we show the mutational constraint of a typical missense isoform. So that's the same line here in these two plots. And we see that the mutational constraint in acceptor regions of near novel isoforms is a little bit more constrained than the typical missense than typical missense mutations in known isoforms. So these are under mutational constraint. And same for same for essential splice sites in the splice donor regions. So these are mutationally constrained a little bit under the value of missense variance. And then lastly, we can look at the mutational constraint in novel isoforms that code for nonsense mutations. And so these are much less mutationally constrained than nonsense variants in known isoforms, but they're still under significant mutational constraint compared to synonymous mutations here. So in general, we do see that these novel isoforms are under mutational constraint. So specifically, like specifically the essential splice sites that are immediately flanking exons in novel isoforms, as well as mutations that code for nonsense mutations in these novel isoforms. For example, like the inclusion of a premature stop codon, so like a stop gain. Okay, so with putting all this together, then, so we have some evidence that these novel isoforms they are under negative selection, and so that suggests that, well, when they're mutated in people, that might then contribute to disease. So like this is all building up to this this use of using this novel isoform atlas to help explain like kind of unknown risk for autism. So basically, like, to answer this question, we're using genome sequencing data that have been collected from ASD families with one individual with ASD. So here in this diagram in black, we have an individual with ASD, and then we have a sibling and parents who don't have ASD. And so by looking at the genome sequence of the affected sibling versus their parents, we can then use this to assess de novo mutations. And so then from these de novo mutations, we can then reinterpret their variants using our new isoform atlas. And we're specifically looking for variants that impact novel isoforms, but don't impact known isoforms. For example, like here in green, we have this this mutation here. And then in the existing isoform atlas, you might not, you might think that this variant is actually in a intron, but then in our novel isoform atlas, we actually see that it's in a new exon. And then if you then sort of predict what the protein consequence of this variant is, you might predict that it actually truncates this novel protein, and then that might contribute to why this person has autism. So to answer this question, we're using genome sequencing data sets from ASD cohorts. So we have three cohorts where we have missing SSC and Spark. And so in total, we have just under 100,000 individuals, 65,000 with ASD, and 30,000 comprising their unaffected siblings. And so this is, this part of this project is a collaboration between my lab and Brett Trost and Mahil Arshad at Sick Kids Hospital. And these Brett and Mahil are human genetics experts. So they're helping us with this aspect of this project. So, so again, looking first at mutations that are impacting known isoforms. So we basically see the expected results. So, we see an enrichment of, individuals who have de novo variants impacting known isoforms in individuals with ASD compared to their unaffected siblings who don't have ASD. And so we have an enrichment, increased odds of having ASD if you have mutations that impact the donor splice sites, acceptor splice sites, mutations that cause nonsense mutations, so basically stop-gain mutations in known isoforms. And so when you combine all these together, again, you have very significant, enrich, having these kinds of mutations strongly enriches individuals for having ASD when you're looking at de novo variants. So these are variants that are just present in the ASD siblings and are not present in their parents. And so then now looking to novel isoforms, when these mutations are impacting novel isoforms, similarly, we see enriched risk. So we see we see increased risk. So for example, for donor splice sites, there's a threefold risk of having ASD compared to, or when someone has one of these kinds of mutations relative to their unaffected siblings. But because because these types of mutations are rare, and because we're not actually looking at that much genomic sequence, then the total number of individuals that have these kinds of mutations, like we're talking 50 people total who have any one of these kinds of mutations relative to over 2,000 years. So we're just not looking at, so while there's an increased risk or while there's an enriched risk, there's we're just under, or I would say this is trending significance for increased risk for ASD with these kinds of mutations. But we're generally heartened that as more and more individuals with ASD are sequenced, especially as more of them are sequenced with whole genome sequencing relative to whole exome sequencing, that we'll be able to use these kinds of novel isoforms to explain more and more risk of ASD as whole genome sequences from more individuals with ASD are collected. All right, so to summarize, here we used PacBio Revio sequencing plus the Kinnex library prep kit to develop an ultra deep atlas of mRNA isoform diversity in developing neurons. We found lots of splice isoforms, many of which are novel. We're really excited or really happy with the quality of the data. Most of our isoforms are supported by corresponding short read data, and hundreds of them also show support at the protein level. Probably among the most exciting parts of this project is just the use of super deep long read data to study alternative splicing. So, and because you have the entire isoform, because you have the entire transcript, you can then ask how changes in that transcript actually lead to consequential changes for the for the whole isoform. So for example, longer UTRs, the transcript, you can infer that the transcript might be likely to undergo nonsense mediated decay and lose critical protein domains or intrinsically disordered region domains. And then kind of one thing that's really exciting about our analyses to date is that it seems like there's fairly considerable coordination between various aspects of the transcript that undergo alternative splicing. So for example, exon usage and polyA site usage, even though those two types of events might be really, really far away. And then lastly, I've showed that these novel isoforms, there's evidence that they're actually under negative selection, meaning that they're, mutations in these isoforms tend not to be present in control individuals, and that contributes a modest degree of risk towards, for having, or knowledge of these novel isoforms can then help interpret mutations present in individuals with ASD. So, lastly, I just wanted to thank my lab. I wanted to thank all the individuals who contributed to this work. This was the work of probably about 10 different labs. And so, my lab has been leading the bioinformatics effort, but we're, heavily in debt to our, experimental collaborators, as well as the patients who helped, generate the resources that we produced. Okay, thanks.
Awesome, guys. So just a reminder, we are now in the live Q&A session. Please use the question box to ask questions that you may have. So I'll start with some that we already have. Srijoy, awesome talk. You had mentioned that, actually, I just saw a question that it will be the first question. Which IPAC differentiation are you using is one of the questions.
Yeah, we are using the NGN2 protocol of iPSC, neuron differentiation. I should just say I'm not an experimentalist. I'm certainly not a cell culture person, but my collaborators who are cell culture people really like this protocol because it's very fast. It generates neurons in a very fast amount of time, say 30 days instead of weeks to months. So it's very, it's just easy to work with. Yeah.
Awesome. So then the next question is also on the experimental design, because you have this neuron development system and you did short read, long read, and proteomics, and we see how they worked together. Why do you what is important about them complementing each other? And if people ask you, if you want to do some similar experiments, would you recommend exactly the same design? Would you have changed anything? What are your recommendations?
That's a good question. I think especially now that we've kind of completed the first phase of the project, as we were we've been working on this data for a while, and as we're putting together a manuscript on this work, one question that we kept asking ourselves is, what's the short read data even for, right? We have I showed you these Venn diagrams, and I'm we were like, why did we even collect it? So I think if you have the opportunity to do super deep long read RNA-seq, then do you necessarily need short read? I don't know, maybe not. And then the question of proteomics, proteomics is uniquely useful in assessing which transcripts are translated. So that's unique and useful, but it still doesn't quite have the depth in terms of ability to assess proteins that are present at low abundances that you would have with short and long read RNA-seq. So I still think that there's something missing. So I wish we had proteomics, but that enabled the detection of peptides that are expressed at much more lower abundances. And I don't think such a thing exists yet.
Speaking of which, one of the questions that mentioned proteomics is, have you considered using Olink's proteomics to validate novel splicing?
Fantastic question. I will tell you briefly, Olink is so funny. They're constantly emailing me to use their stuff. And I'm like, guys, stop emailing me. But that issue aside, my understanding of the way Olink works is it uses antibodies that are then conjugated with some nucleotide barcode sequence. So based on my naive understanding of that, then you would need a antibody that's kind of custom for your novel isoform, right? Yeah. So your antibody, the peptide sequence of your antibody that needs to span the region covered by your novel isoform. So if it doesn't do that, then Olink isn't quite appropriate in this way, and so generally, I think antibody-based methods, because they would be designed against canonical or known proteins, then they're just not as amenable as more data-driven methods of finding proteins like mass spec-based proteomics.
Got it. So then moving on to the next question, which is very in line of continuing on the do we need short reads if we have really deep? You had actually asked a question in the webinar. I was like, I think this is the deepest. Is it? Liz will answer this question. I would say that it should be one of the deepest. We did just put out a WTC 11 preprint that just slightly beat you, just slightly. I think we had an average of 40 to 50 million reads, and I think yours were 30 to 40. So we'll just say you got it too. Where we will not try, we are not trying to compete for those numbers. But a great question from one of the attendees is, did you actually run one smart cell per sample? And that, would you still recommend that? Because I think going deep is that you can ask the question like, did I need to go that deep to get the answers you were looking for, such as differential expression, proteomics validation?
Yes. Okay. So, So why did we run one sample per smart cell? Grace, our PacBio rep, said that this data was generated pre SPRQ. Again, I don't know what that means, but I think
it has no meaning. Just sparkly, great chemistry.
Okay, whatever. So, why did we do that? I think we didn't know how much depth we needed, and that's the honest answer before we collect the data. Like, looking back now, why did we collect such deep data? I don't regret it. I certainly, as a bioinformatician, I do not regret collecting such deep data. We used our data for two purposes. The first purpose was generating this super comprehensive isoform atlas, right? So depth enabled us to find a bunch of isoforms and then be somewhat selective about what we were calling an isoform versus not calling an isoform. So depth is important for that, but it's actually not as important as for the second purpose, which is differential isoform usage.
Right.
That needs super deep data. You can't do that if you just have like 1 or 2 reads per isoform. And so I think if you're especially, this field has to be moving towards differential isoform usage. Like, of course it is. And so I assume that, well, anyway, depth is necessary for that. Like, short, yeah. Sure. But if all you care about is just isoform discovery, then I don't think you need to go that deep. And 10 million reads is probably fine.
Lots of questions coming in. I'll try to do my best to get through them. Have you used any AI-based predictors of sequences for cryptic splicing events? And if so, have you seen them to be enriched in novel exons?
Yeah, fantastic question. We've actually never thought to do that. My collaborators, Brett and Mahil, are actually using, they're using tools like, what's the big one called? Splice AI. They're using Splice AI to try to find kind of genomic sequences that are kind of, So the way those work is that they basically find places in the genome that might be kind of contributing cryptically to splicing. And then that kind of that's like a different way of getting a reference resource of which regions of the genome are especially important to splicing. Whereas like our way of doing it was like this kind of data-driven way, or like this, like literally just using data from neurons to figure out where are they spliced. A different way would just be like to use like genome sequence data and no RNA data, whereas like ours was RNA only. But
right.
So then you have like, then you can use these tools to figure out what regions of the genome are especially might be involved in splicing, and then you can go looking for mutations in those regions as well. I think they are working on that in parallel, and I think they find some enrichment of regions of the genome involved in cryptic splicing that are enriched in ASD, but long and short, they're pursuing that in parallel. But we haven't actually thought to do that in this project. But the other part of Alan's question is, are these sequences preferentially found in novel exons identified by long recency? We haven't actually looked, and that's reasonable. We should look.
Yeah. Here's a coming back to, I know you can see the questions too, a little further back in the earlier questions. How do you account for sequencing bias? I'm trying to interpret this question. How do you account for a sequencing bias when performing either differential transcript usage or differential transcript expression? I'm trying to, if you understand what the sequencing bias means here. Yeah,
I think what they mean, what that must mean is just probably about read depth. So I think like relative to the data sets I've worked with, our read depth surprisingly isn't that bad. So it's like imagine we had one sample with 1 million reads and another sample with 100 million.
Right.
Then how would you account for how would you do like your statistics appropriately when you have like 2 orders of magnitude difference in read depth? I think in our case, our read depth for our lowest sample was 25 million reads and the highest was 40 million. So thankfully, just from a data collection standpoint, I don't think read depth is a big issue, but To be honest, like I think I would just hope that this issue is kind of managed by the tools that we're doing for this analysis. We're using Isoform Switch Analyze R. Again, I didn't come up with that name, but that's a common tool for these types of things. And so I would hope that they have some built-in way of addressing, some sequencing bias related to, like, say, for example, read depth.
Yeah, so I actually great that the person actually corrected what his sequencing, but they meant length bias. So Kinnex bias is towards 1 to 2 kb fragments. You might miss out on longer reads more often. So, I think what they're saying is, let's say if you have a 5 kb transcript, a 10 kb transcript, is it likely that you're not going to capture the differential expression analysis because of that?
Fantastic question. I don't think I ever really thought about this, but let me try to think it through on my feet. So, okay, first let me say that, like, I don't know why, but our data is something Liz and I talked about when we first met, like probably 6 months ago, talking through this data. She was like, "Why don't you have more ISM transcripts?" And
yeah, I did. I did ask that. Why is your data so great?
Yeah, so I don't have a good answer for that, but I think thankfully our transcripts don't appear as fragmented as some of the other data sets we've looked at. So thankfully, I don't think that would. Okay, so that's a good thing. Then on the other side of the question, then let's imagine like shorter reads or shorter transcripts would tend to be sequenced more. If you sort of put your short read hat on, shorter transcripts will be sequenced more, and then you might see them expressed more in certain contexts. I think when we've manually looked, we haven't seen strong evidence of that, but I'm sure if we look more, like we might see some evidence of it.
One of my strange hypotheses might be that you had such a clean sample because you're using, basically a generated neuron. You're not using like a lot of times in brain samples, they're, from, post-mortem samples, and they're really degraded RNA. So, I'm used to seeing things that might be coming from like a wider range, which the ISM might be both an effect of RTs, but also just far from degradation. And perhaps, you got very clean samples because the RNA was so high quality.
Yeah. I completely agree, and I concur. And what Liz said is also consistent with my own experiences looking at a bunch of brain postmortem data sets as well.
Yeah. Okay, I think we can get through 2 more questions. This is a different topic. It's very interesting to see the UTR changes in your data, and it complements the idea that neurons use 3' UTRs to perform directed or compartmentalized translation. Can you observe these changes on a cell-to-cell based difference in your system, or is a readout from a collection of neurons together? Can you tell them apart even?
Okay, so I think the question is basically kind of about, I think when I read this, I interpret this as bulk versus single cell.
Yeah, that's why I read it too.
Right, and so, I should say that I'll just say again, we collected this data from bulk samples. And so one convenience of this system is that it's fairly well characterized, and at the iPSC stage, it's mostly all iPSCs, and at the neuron stage, it's mostly all neurons or excitatory lineage neurons.
Wow.
Is there heterogeneity within those cells and systems? For sure, there is, and but I think this system is like I feel fairly comfortable doing a bulk-based analysis based on this. So, but for sure, can you imagine that, like, let's say one type of cell is expressing one isoform and another type of cell is expressing another isoform? Sure. And I look forward to like seeing more single cell long read data sets to tease that out more in the future. Right. But I think that will need to happen later, obviously, that'll happen later.
Awesome. Okay, I'm going to do one last question. Hopefully this is a quick one. What is the minimal number of reads or assembled transcripts that you use to define an isoform? Like, what's the cutoff?
Yeah, for us, we can call an isoform with 15, at least 15. And for us, that means at least 5 or more unique transcripts in at least 3 samples. So yes, it's super arbitrary, but by God, like this field, as like not a long read person, like I was kind of shocked at what other people were the evidence that people were using to call unique isoforms, and I think it's helpful to be somewhat stringent. And I think, could we be even more stringent? Yeah, for sure.
All right, awesome. Let me see. I think we are good. Thank you so much, Srijoy. This was a wonderful talk. Thanks to all you attendees. Please fill out the survey. We really would appreciate it. And you'll get a recording in your email soon. And thanks for all the emojis. I love this emoji option. All right.
Thank you.
Thank you, guys.
Thank you, everyone. Bye.
Bye.